Hello, welcome students. We shall start the next nucleic acid, that is ribonucleic acid. That is RNA. You know that there are two types of nucleic acid. One is DNA, next one is RNA. About DNA we have studied. Today we shall start the ribonucleic acid. Now, that is ribonucleic acid is also a polymer of nucleotides. There are so many nucleotides combines to form the ribonucleic acid. Understand? Now, the nucleotides, but there is somewhat differences between the DNA and the RNA. Now, it also contains sugar, nitrogenous base, phosphoric acid. All the three things are present in that. Understand? But hey, in the DNA we have seen that the sugar was deoxyribose, but in that it will be ribose. And we know that the deoxyribose a pentose sugar, first, second, third, fourth, and this is the fifth. H in the deoxyribose, second and third position contain hydroxyl, but in the deoxyribose only on the second position there is hydrogen. So the fundamental difference is that the second position contain OH and here is contain H. So here in the ribose sugar, two hydroxyl groups are found. So it is more reactive, means that is contains two functional group, two OH. Now this is the first position, second, third. Here is nitrogenous base is attached. This is the fifth position. Here is phosphorus is attached. Understand? The phosphoric acid is attached. This is called glycosidic bond, and this is called phosphodiester bond. About this we have already studied in the DNA. Understand? So that is the sugar base is attached to nitrogenous base on one side, that is by means of glycosidic bond, and the other side the fifth C is attached with phosphoric acid, that is phosphate, by means of phosphodiester bond. यानी नाइट्रोजन जो पहले पोजीशन पे होता है यानी राइबोज के फर्स्ट पोजीशन पर जो कार्बन होता है उसके साथ नाइट्रोजनस बेस अटैच्ड होता है, समझे? दैट इज नाइट्रोजनस बेस अटैच्ड होता है, और जबकि पांचवें कार्बन के साथ फॉस्फेट या फॉस्फोरिक एसिड अटैच्ड होता है। ये फॉस्फोडाइस्टर बॉन्ड से अटैच्ड होता है, जबकि नाइट्रोजनस बेस ग्लाइकोसिडिक बॉन्ड के साथ अटैच्ड होता है। तो यहाँ शुगर राइबोज था, जबकि डीएनए में डीऑक्सीराइबोज था। तो एक मेन डिफरेंस ये हो गया कि द शुगर इज फर्स्ट वन, मीन्स दैट इज डिफरेंस बिटवीन फर्स्ट पोजिशन इज दैट डीएनए कंटेन डीऑक्सीराइबोज and this contain ribose, both pentose sugar. This has 2 hydroxyl group and here is one less. Now the next difference, the first difference between that sugar, DNA, deoxyribose, ribose. Second difference, the nitrogenous base in the DNA. We have seen that the nitrogenous base adenine, thymine, guanine, cytosine. These are the nitrogenous base which are attached by hydrogen bond. This is hydrogen bond. So nitrogenous these are, but in case of ribonucleic acid, that is instead of T there is U. Instead of T, GC. So the base of the ribonucleic acid is AUGC. When in place instead of T means thymine, there is uracil. So that is the second difference in a nitrogenous base. Means here is, so the nitrogenous base में भिन्नता होती है. In place of thymine there is uracil in the. तो first difference होगा, second difference ये. Third one, the main difference is the deoxyribonucleic acid is a double helical structure. We have studied. This is a double helical structures. But double helical or double stranded. This is double stranded structure, but this one is single head. That is a single, although there is some modification which we shall see in the next. Then what we see, first one is difference between sugar, second difference between the nitrogenous base, and third difference between the strand, that is double stranded, and this is single stranded. First, next one, there is no type of DNA. There is no type of DNA, means DNA no types, understand? But although you have seen A, B, C, that is another things, but ribonucleic acid it is of 3 types on the basis of their function. On the basis of function, there is division on the basis of number of nucleotides. Understand? You have seen In this DNA there is also division A, B, Z like that. A, B, C, D, Z like that you have seen, and that on the basis of their number of nucleotides, one complete trial. But here is, that is on the basis of nucleotides in each trial. That but here is on the basis of function is the basis on which ribonucleic acid is divided. Next one, first one is what are mRNA. These are the three types. Now M stands for messenger RNA, R for the ribosomal, and T for the transfer RNA. It is also known as sRNA, that is soluble RNA. We will discuss in the other time of this topic. So these are the three: messenger, ribosomal, and transfer RNA. These are the three types. We know that. Let's see. This is a cell. This is nucleus. It is a chromosomes on which DNA is present, and in the cytoplasm this is ribosome. On the codes or information starts at the DNA. Understand? Starts about the code for the formation of protein is initiated as the DNA which is present inside the nucleus, but the ribosome and the protein is formed on the ribosome. So how the message about what type of protein will forms, it will comes from DNA to ribosome. हम देखते हैं, जानते हैं कि न्यूक्लियस में क्रोमोसोम्स पाया जाता है जिस पर डीएनए होता है। प्रोटीन सिंथेसिस कहाँ होती है? राइबोसोम में, जो इन द साइटोप्लाज्म पाया जाता है। तो जो यहाँ जो इंफॉर्मेशन होता है प्रोटीन्स के बनने का, वो डीएनए के माध्यम से दिया जाता है। वहां एक कोड बनेगा और उस कोड को यहाँ राइबोसोम्स पर भेजा जाएगा कि किस टाइप का प्रोटीन्स बनाना है। समझे? तो अब ये क्या हुआ कि अब यहां से यहां तक जो मैसेज को, ये उस मैसेज को ले आना है, दैट इज द वर्क ऑफ, उस मैसेज को ले आने वाले को हम लोग क्या कहते हैं? मैसेंजर आरएनए। यानी ये बनता तो है न्यूक्लियस में, लेकिन इसका फंक्शन होता है इन द साइटोप्लाज्म। इट ब्रिंग्स द मैसेज फ्रॉम द डीएनए टू राइबोसोम, दैट्स व्हाई इट इज नोन एज messenger RNA. When it brings the message from the DNA to ribosome. Next one, ribosomal protein RNA. That is, it founds on the ribosome and helps in the formation of protein synthesis. Means the protein synthesis is synthesized by this ribosomal RNA. Next one is tRNA. It is, so we know that proteins are made up of amino acids. Understand? And the amino acids are found in the cytoplasm. प्रोटीन एमिनो एसिड का बना होता है, और एमिनो एसिड्स कहाँ पाया जाता है? साइटोप्लाज्म में पाया जाता है। समझे? अब अलग अलग प्रोटीन्स का अलग अलग एमिनो एसिड्स, एमिनो एसिड्स तो जानते हो कि 20 टाइप्स, और उसमें से कौन उनका सीक्वेंस कैसे होगा, कैसे सजाया जाएगा तो ताकि वो प्रोटीन बने, वो तो अब डीएनए से वैसे ही हो। तो अब उसको इकट्ठा करना, समझे, ये कौन सा एमिनो एसिड आवश्यकता है इस प्रोटीन के लिए, तो उसको इकट्ठा करना और उसको ट्रांसफर करना राइबोसोम के पास ताकि वो प्रोटीन बनाए. दैट इज द फंक्शन ऑफ टीआर, ट्रांसफर मीन्स इस ट्रांसपोर्ट द एमिनो एसिड. दोस्तों ये क्या हो गया, मैसेज ले जाना, यहाँ जो है प्रोटीन सिंथेसिस में हेल्प करना, और ये transfer amino acids from cytoplasm to राइबोसोम, यानी साइटोप्लाज्म से ट्रांसफर करना. पहले साइटोप्लाज्म उनको चुनना, सेलेक्ट करना कि कौन सा प्रोटीन, कौन सा एमिनो एसिड उस प्रोटीन के लिए चाहिए, और उसको पहुंचाना, उसको ट्रांसफर करना. हम यानी उसको ट्रांसपोर्टेशन ऑफ एमिनो एसिड इन अदर वे भी कर सकते हैं. तो इस एमिनो एसिड को ट्रांसफर करना वहां, दैट इज द फंक्शन ऑफ टीआरएनए. तो ये तीन टाइप्स के हो गए. वन इज मैसेंजर, नेक्स्ट वन इज राइबोसोम. So now we shall discuss each one by one. First we shall discuss the messenger RNA. Now we shall see that formation of ribonucleic acid, that is RNA. This is come from the DNA, you know, and this process is called transcription. How this forms, the application we have already studied in the last week. Here is a transcription. You see that, you know that this is the DNA strand. It falls bifurcates. Understand? This is the bifurcates, means the two strands get separated. Now that this separation, these are called template. This is the template, and this is also the template. Understand? Now these templates undergo replication and forms DNA. But in case of ribonucleic acid, only one template takes part, not two. One thing you must listen. In the formation of ribonucleic acid, only one template takes part. Understand? On the there, and you know, we know that A, T, G, C, these are the unit of DNA. Understands? So the template contains the same way A, T, G, C. Understands? So these are now these templates. Now it will form its complement. In case of DNA, T will form A will form T, C will form G, G will form C, as usual. But if it is so, it is the DNA. For DNA, this template will, this complement will forms. But in the ribonucleic acid, the template will form somewhat different. AU, understands? AU, this is T will not found in that. Understands? Acha. G, sorry. So the complexity for the AUUAGC means only one template will take parts. Understand? So why if you there is a question that why one template takes part. Understand? उन्हें पूछो सवाल उठता है कि फिर एक ही टेम्पलेट क्यों भाग लेता है, हम ये दोनों टेम्पलेट क्यों नहीं भाग लेते हैं राइबोन्यूक्लिक एसिड के बनने में। यदि दोनों टेम्पलेट भाग लेंगे तो इससे 2 राइबोन्यूक्लिक एसिड बनेगा और 2 राइबोन्यूक्लिक एसिड बनने से एक डीएनए से 2 तरह के मैसेज जाएंगे प्रोटीन बनने के लिए, जिससे प्रोटीन बनने में देयर इज डिसऑर्डर उत्पन्न होगा। तो एक बनता है इसलिए कि 1 डीएनए सेंड 1 मैसेज 1 ऑर्डर्स टू फॉर्म द 1 टाइप ऑफ जीन। अंडरस्टैंड? So अच्छा, and that's why it is because it is formed by single template, so ribonucleic acid is single strand। एक तो सिंगल टेम्पलेट से बनने से वैसे एक होता है, अगर मान लें एक डीएनए से 2 तरह के राइबोन्यूक्लिक एसिड बने तो 2 टाइप्स के मैसेज जाएंगे, 2 टाइप के मैसेज जाएंगे और 2 तरह का प्रोटीन बनेगा। तो एक डीएनए से 2 मैसेज जाने से 2 प्रोटीन बनेगा। तो जो प्रोटीन के सीक्वेंस में प्रोटीन के ये में दैट विल बी डिसऑर्डरनेस आएगी, समझे? दैट्स व्हाई ओनली 1 टेम्पलेट टेक्स पार्ट इन द ribonucleic acid formation. Now we see how it forms. Suppose now we see how the DNA changes to ribonucleic acid. Now suppose if that DNA strand, its base is A, T, A, G, C, T, A, C. This may be the base. In the DNA component, if it is DNA, it will form T, A, T, T, C, G, A, T, G, A, T, C. It is the normal in DNA. Understand? But in place in the RNA, what it will be forms. Understand how this will take part in the formation of ribonucleic acid. But if it is the RNA, if it is the ribonucleic acid, the basis will be U, A, U, C, G, A, U, C, A. Means in place of thiamine is replaced by uracil. That is the ribonucleic acid. Now we see how the initiation of ribonucleic acid takes place. How the ribonucleic acid is formed from the, suppose this is the DNA 5 prime 3 prime, that is the DNA. Next, how it will form is change into the ribonucleic acid. Means next this DNA it will separated into many parts, some coding parts and some non-coded parts. Suppose these are the coded part, these are coding part, these are the non-coding part, coding part, non-coding part, coding, non-coding, coding, non-coding, coding, non-coding, coding, non-coding, this is non-coding. In the same way, that is coding and non-coded part. The coded part is called exon and the non-coded part is called intron. So that is coded exon, intron, exon, intron, exon, intron, exon, intron, exon, so on. Understand? Now they are divided into code and non-code part. Now the splitting takes place. A splitting, if there is coding and non-coding parts. A splitting of this coded part will take place. Understand? That is done by a splicing, and that takes place by endonuclease enzymes. Then each parts will be get separated. This is the exon, intron, exon, intron, exon, intron, exon, and so on. This will become fragments, and these are called Oczynski fragments. These are called Oczynski fragments. Now their joining will take place, and joining of exons will take place by ligase enzymes. By ligase enzymes, the joining will means all the exons are, all the coded part will they are all the exons. Suppose exon 1, 2, 3, 4, 5, 6 like that. And that takes place by means of enzymes ligase, the addition of this exon. Now, but this exon is an unstable filament, unstable compound. Now, so there for stabilizing it, there is capping and tailing takes place. There is capping and tailing takes place. Means formation of cap and tail. Understand? How the formation of cap and tail? Capping. They are the exons. Understand? Now, because it is an unstable, so a cap is formed on. This is 5 prime, this is 3 prime. a cap is formed on that on site, and a tail is formed on this side. Understands? After formation of cap and tail, they bind each other and so it becomes established. Understand? And this capping, this is contain methylated guanine, and this contains so many amounts of adenine. So polyadenine. So these are and so it's the main part is now divided into this part, this part. This is called promoter. This is terminator, and in these totals are a structural gene. So that structure is formed, and it is the structure of messenger ribonucleic acid, and the process is called termination, and the process is called transcription. Understand? So now in this way messenger RNA is formed.